Awesome. All right. Hi, everyone. My name is Aya, and I'll be your moderator for today's session. On behalf of the Kazakh International Science and Technology Association, I'm delighted to welcome you to our Guest of Honor seminar series. Here's what to expect today. We'll begin with welcome remarks from Dr. Halmjan Isabekov, the founder of our association, and then we'll move on to the keynote talk by our distinguished guest, Professor Shoukhrat Mitalipov. Finally, we'll open the floor for Q&A session. If you have questions at any point, feel free to type them into the chat. We'll be collecting them throughout the session and addressing them during the Q&A. Today's conversation goes beyond the science. We are here to explore the challenges faced along the way, the key moments of insight, and the meaningful questions that continue to shape this evolving field. We hope this session sparks curiosity and thoughtful dialogue. I would like to give a word to Dr. Halmjan Isabekov.
Good day, everyone. My name is Halmjan. And first of all, I want to say thanks to Shoukhrat Agha that he accepted our invitation, and we very delighted to have him today. So before he starts, I will introduce after my talk. So I want to briefly touch about our association. It will take 5 minutes. So our association is NGO association. It's not government association. We have more than 80 members, including PhD, postdoc, and professors from Kazakhstan conducting research in world institutions. So we established in 2014, and we have very dynamic and welcoming community. So shortly about our team, our president is Sergey Mirmanov. He's the president of Japanese private Vice President of Japanese Private University, Ritsumika University in Iceland of Kyushu. Unfortunately, today he is not able to attend because it's very late. And so about myself, I'm Halmjan, founder and deputy director for international relations. Aya, today's moderator, secretary of our association, and we have board member. Here we only have Juan Shiraliyev. He is a former vice minister of education and science in Kazakhstan, currently professor in Nazarbayev University. So, and we also have other team members. And about our association, mission of our association is maintain, strengthen, and develop common intellectual and cultural space of Kazakhstani scientific community across the globe in order to provide expertise to professional community and public on scientific and educational discoveries. About the strategy is to mainly initiate and coordinate collaborative projects in science and education with Kazakhstani organization and with international organization as well. So our president of our association is also a member of Science Совет ученых при президенте. So we knowing what's happening in Kazakhstan, and we're trying to be in line with strategic purposes in terms of science development. And our association has been more than 10 years. So we organize more than 35 online and offline meetings of Kazakh researchers working around the world: China, Japan, United States, UK, Hong Kong, New Zealand. So as you can see on the left side, there's a panel discussion which we often organizing on different topics. We're inviting several speakers and try to find the topics that mostly interested by our community. It's mainly for young scientists. And second one on the right side, you can see there's the guest of honor speaking a seminar series, which today we have Professor Mitalipov and others which have been invited before from different countries. And last one, we also invited a flight surgeon, doctor of Japanese astronauts, my friend, best friend, Kazuhito Shimada. So we're trying to find different, very interesting individuals with very much experience and try invite him there and share the experiences with us. Yeah, and what's also important, we have this project of 100 Kazakh alum. It's prominent researchers originated from Kazakhstan, but currently working around the globe in different countries. This project leading by Juan Shiraliyev, where he interviewing these researchers. So if you're interested, you can go to our website to read their interview. And this all recordings is uploaded to our YouTube channel, so you can watch them. We also had session with Sayasat Nurbek about current situation and strategies of Kazakhstan in science, and trying to be in line with them. Other than that, we also organizing panel discussions, which is relevant to Kazakhstani situation and citizens. So last year we had this big flood, and we organized this emergent meeting with foreign and Kazakhstani scientists in order to give some recommendations, scientific-based recommendation. So yeah, additionally, we also work with international organizations like Takeda Science Foundation. This is kind of Japanese Nobel Prize organization which award Japanese scientists and also encourage foreign scientists come to Japan to do research. And we organized Central Asian and Japanese fellowship opportunities for medical doctors and researchers, try to invite this organization to Kazakhstan and share what kind of opportunity they have. And every year they choose 10 candidates around the world in order to come to Japan and do research and clinical training in a Japanese leading institution. Yeah, and we also try to select candidates for a Japanese PhD scholarship, as you can see on the right side, MEC scholarship. Yeah, and we collaborate with other association, as we have Kazakh Association of PhDs in North America, Kazakhstani universities, and Ghalem project as well. So it's very important, and we currently doing very interesting projects. So we will keep update our members. And what's interesting during Saisat Nurbek's interview, he said currently a lot of foreign institution building universities and campuses in Kazakhstan. As you can see, there's KAIST University from North Korea, Cardiff University, Heriot-Watt, and they building these university campuses in Kazakhstan. So we're trying to also to be in touch with them in order to understand what kind of necessities they have and try to find positions maybe for our members. Yeah, and lastly, yeah, this interactive map on our website where you can find the Kazakh scientists around the globe and connect with them. And last one, we had this year a summer retreat in Bavarian Alps for our members. If you're a member of our association, you will enjoy all benefits that we have. That will be 3 days of enjoying traveling and academical meeting, some kind of, and yeah, we will have this summer. And if you're interested to become a member, please go to our website, kesaeurope.org, fill it up and send it to us, and we will share with you all benefits that you have as a member. So so I'm ending with this, and if you're interested to join our Telegram channel, please scan this QR code and visit our website. So thank you very much, Aya. Floor is yours.
Thank you, Ramjan. The Kazakh International Science and Technology Association is proud to welcome Professor Shoukhrat Mitalipov, a global leader in stem cell research and germline gene therapy. From his early days in Kazakhstan to his pioneering work at forefront of reproductive biology, Professor Mitalipov's career has been nothing short of extraordinary. He's especially known for developing a technique to prevent the transmission of mitochondrial DNA diseases from mother to child, a major milestone in modern medicine. His lab's creation of human embryonic stem cells from skin cells was recognized as a top 10 scientific breakthrough by Nature. Professor Mitalipov is the director of the Oregon Health and Science University Center for Embryonic Cell and Gene Therapy, and a professor at the Oregon National Primate Research Center. After earning his PhD and completing postdoctoral training in developmental biology at Utah State University, he joined OHSU in 1998. Since then, he has received numerous accolades, including Nature's Top 10 People Who Matter in Science and China's prestigious Thousand Talents Plan Award. Today's session will blend scientific insight with a personal journey, offering a rare opportunity to understand what drives innovation and how it can shape the future of medicine. Please join me in welcoming Professor Shoukhrat Mitalipov.
Thank you, Aya, and thank you, Galamjan, for a nice introduction and the opportunity to talk. Okay, let me figure out how to share my screen. Um, yes, my presentation. Trying to find it again. Documents. Okay. So I'm gonna do a little talk about, I guess, myself a little bit, but then mostly I'm gonna talk about, I guess, science. So I'm still trying to share screen, Windows. Oh, sorry. Okay, here's my presentation, I guess.
Yeah, we see
You
you.
see? Okay. Um, yeah, so let me go back to beginning of my talk and go to. So again, so mostly I'm gonna probably start with talking about about science, but a little bit about myself. As you probably know, I am of course from Kazakhstan. I born and grew up in Almaty region. So my dad was a teacher in high school, so he kind of moved a lot, but mostly in the Almaty region. So and I graduated from high school in 1970. I think it was 78. Yeah. And then I served in military. It used to be kind of drafting, supposed to be in military for 2 years. And I served in Belarusia. It was 1979 through 81. And after I was released, I actually went straight to, I think it was fall, when I came to Moscow, and I entered into Timiryazev Academy. It's agricultural, as you know, school where I graduated in 97. It was kind of, I guess, master degree, and primarily I was focused on reproduction. But by specialty, I am a zootechnic or zooengineer, whatever they call. I don't think that specialty exists anymore. But I when I graduated in 97, I actually worked for I think 2 years in Russia. I was главный зоотехник в колхозе. So that was a kind of very different path in my career. But then I decided that I like science, and I was always drawn to science, and that's why I entered into PhD program in Research Center for Medical Genetics in Moscow. So I entered in 19, let's say 89, and then in 94 I graduated with PhD in developmental and kind of genetics, but primarily I was studying clinical genetics, and that's why I'm interested in clinical genetics, particularly inherited gene defects that affect the humans since then. Once I graduated, and as soon as I got my PhD diploma, I actually went to postdoctoral training in Utah State University. Ghalamjan is mentioned, actually, if I forgot very briefly, since I got my PhD diploma in 95, I actually went to Germany for postdoctoral training in University of Münster. But I think when I was still at the PhD in Moscow, I applied for a kind of fellowship trainee grant in United States. And I won it. It was in 95. So sorry, it was 95. So that's why I went to Utah State University. So I did my postdoctoral training in USU. It's actually located in small town in north of Utah, Logan. And then I went to Oregon. So it's a Portland area. And initially, I started as a staff scientist in Oregon National Primate Research Center, which is one of the, I guess, 6 or 7 large primate research centers in the United States. It's actually part of the Oregon Health and Science University, which is kind of big medical school in Portland area. So my career was a staff scientist, and I think it was, it was in 2000, I believe it was in 2006 when I became assistant professor, and since then, I kind of was promoted now full professor. In 2013, I founded the center, which we call Center for Embryonic Cell and Gene Therapy at the university. So it's a kind of standalone research center, part of the university, where I am now as a director and a professor. So the center is actually, as you could see by its name, primarily we started working on gametes and embryos, and that's why it's called embryonic. But the goal was not just to do basic research, but primarily try to develop therapies, and we kind of divide them into 2 kind of different areas, cell and gene therapy. So some of these projects I'll be presenting you today. So Kalamjan sent me a few general questions about how I, came to science and key mentors in my career. And I would like to say, actually, I was very lucky throughout my career. I met so many wonderful people who contributed to what I am today. But I'm going to just mention probably a few. So first of all, it's Vladimir Ilyich Ivanov. He was my PhD mentor when I was in Institute of Медико-генетический научный центр. I think that was name of the center at that time. So he was a director of that center, but he was also he had a lab which is used to call Laboratory Генетики развития. So I was his PhD, and he was one of the first, I guess, my scientific mentors who helped me a lot to understand science and how it works, and particularly area of the research, this developmental genetics, which I found fascinating. I still am. So I had, of course, lots of other mentors, but I would like to mention Don Wolf. So he's a professor. He's still, kind of emeritus professor at the OHSU here. But I came in '98 to his lab. I joined. He was at the Primate Center, has a research lab, and I came as a. Initially, I came as a staff scientist, but then I become, assistant professor and so on. So he was instrumental in helping me to become independent professor and set up my own lab and so on. But he was also one, when I joined OGC in '98, he actually had a research lab at the Primate Center, but he was also established this in vitro fertilization clinic at OHSU. And so because he was running kind of clinic and research lab, so when I came, I also worked part-time at the clinic under his direction, of course. And that's how I learned clinical in vitro fertilization area, and which has now become my primarily clinical, I guess, area where I develop treatments and introduce into this area. And I guess one of the people who helped me with my career was Keith Campbell. Not many people probably know it, but he's the one who actually created, if you remember, this clone sheep dolly. It was in late '90s, and when I came to Utah State University, I went to one of the meetings and I met him. So he become actually he was my friend, but also mentor, particularly in area of this cloning and reprogramming, rejuvenation, so we were, unfortunately he passed away, but this was probably one of the big area of the cloning that I become interested since I met Keith. Let me see. So the book that had a significant impact at my career, actually probably was lots of different books I read, but I would think that the book by Anne McLaren. So she's a very famous scientist from UK, and she's actually a pioneer in developmental biology and particularly in vitro fertilization. She wrote, of course, many books, but the one book which is called Mammalian Chimeras, at least that's in English. This was translated, I guess, in Russian, and I read it, I think, when I was still in Timiryazev Academy. And that's how I become drawn to this the area of experimental embryology, and I entered into PhD program with her book. I actually was lucky to meet her in person, and that was, I think, in 2008, and that was one of the conferences in UK. And so I was invited speaker there, just like today, and she was there too. And I remember we were sitting with her on one of the receptions, and I told Anne that, Anne, I came to this area because of you. And I said, I read your book. And she was very impressed. She says, which book? I said, I think in Russian it's called Mammalian Chimeras, and apparently she had it. And I said, oh, I wish I had this book with me so you could sign it. Of course, I didn't have that book with me, but she kind of grabbed, there was a kind of invitation letter and wrote there that, for Shoukhrat, was all the blessings in science. And I actually have it, framed. It's in my office. It's one of the, I guess, my greatest books. And she unfortunately, she died. When I met her, she was already kind of, I think, in her 80s, but she had an accident, I think, 2 months after I first met, and died. So that was, my book, and particularly her career was greatest inspiration for me. And I've been doing actually chimeras in my research since then, and I'm still doing it even today as we speak. We have research projects when we use this greatest technology when we can make chimeras. But anyway, so going back to research, focus of the Center for Embryonic Cell and Gene Therapy. Of course, we do basic biology research, and one of the area that they've been always interested in lifespan and aging and what controls it. So that's, I guess, going to be topic of my few slides later. But I was always interested in gamete, that means sperm and egg, and early embryo, right after fertilization, and also pluripotent stem cells. As you know, you could establish a kind of cell line from early embryo. It's called embryonic stem cells, and that was one of the area I was working at my PhD diploma. I become interested in mitochondrial genome, and I'll tell you a little bit why a little bit later, but it's one of the, I guess, major generic focus. Because of, it's interesting always study, but we need to make sure that, especially in clinical area, you have to focus on developing therapies, and that's why majority of projects in my center, in laboratories, is therapy. So cell therapy is we actually work, as I said, with eggs and sperm, and that's why we have many projects. Particularly, I'm focused more on egg rather than sperm, just so the oocyte regeneration, and we have a project so we can make them younger or improve the quality by cytoplasmic mitochondrial replacement. So the area of that research is called MRT, mitochondrial replacement therapy. And then that was a kind of one of the earliest projects, but now we're working on more advanced therapies. So basically, today's IVF or in vitro fertilization, it's actually assisted reproduction that can help with fertilizing eggs and sperm and creating embryo at early stage in vitro. And as you know, there are maybe more than 10 million children born. That includes even in Kazakhstan, just thanks to IVF. But the IVF, fortunately, only works if you have, mature and functional gametes. So all we do is just to fertilize them in vitro. But majority of the diseases, infertility conditions due to the lack of gametes, and an idea that we can create eggs and sperm from, say, other cells of the body that patients have. In this case, it could be just blood cells or just skin cells. So that area of research so far, it's called in vitro gametogenesis, and it's generating haploid, cells from somatic cells, which are normally diploid. And we actually do it by technology. It's called somatic cell nuclear transfer, and I'll be telling a little bit about that. Because, I was primarily trained as clinical geneticist, so that's why I work with the patients that carry mutations inherited from parents. And since my clinical area is in vitro fertilization, so we can actually now access to eggs and sperm or early embryos in vitro and produce embryo in vitro. So it gives us opportunity to correct the mutation at that stage if patients or parents have pathogenic mutations. So it's an area called germline gene therapy. We now, of course, use gene editing to correct mutations, but we also use some other techniques like mitochondrial replacement, because the mitochondrial genome is very hard to correct. It's basically replacement is easier. And the other areas, gene therapy, is mitochondrial transfer, because it's as organelle for regeneration or rejuvenation for many age onset conditions, and that relates to eggs, but also to somatic tissues. So going back to my interest in development, as you know, we all start as egg and sperm fertilization, and the first cell we call it now the zygote. And once fertilization starts, I think the development starts progressing. It's kind of one way, development, and you cannot stop it. You cannot, pause it, unfortunately. And as I mentioned, I particularly study a very, very short period in development, which is called pre-implantation. It only lasts 5 days, at least in humans, as you could see. But then, the embryo at day 5 will have to implant and becomes a fetus. So we all know that, this development is about 9 months. We develop usually in uterus, and then of course after we're born, the rest of the stuff and so lifespan of particular humans is a kind of set, and of course it could be plus minus, but on average, I think I looked up in the Google in humans, it's about 73.5 years. And I was always interested, why, for example, we have to live, say, 70 to 80 years. But as you know, there are different species that could live actually different lifespan, like, some insects that could just live just a week or two. I study mouse, of course, as a model. I think they live around 2 years. It could be maximum 3, but I think the 2 in the laboratory mouse is usually is a limit. Different species have a little bit longer, and I guess there are some sharks. They could live 400, and some other organisms, like you probably you remember in biology we study hydra, and apparently hydra lives forever. It just doesn't die. Anyway, so my interest was, what is it makes, every species live a certain lifespan. And I always thought it's probably encoded in our genes. I thought, when I was kind of just entered into PhD program, I thought, there is probably some kind of master gene in our genome that actually, predetermines how long we live. And that's why, there is nothing you could do about it. And when I entered the PhD program, I remember the sequencing of our genome just started. I remember it was a human genome project. And I was waiting, for results. And I thought, we're going to find that master gene that actually predetermines how long we live. And I remember I talked to my supervisor, Vladimir Ilyich Ivanov, saying, I think I'm going to find this master gene. I remember he kind of said, "Дерзайте." That was his word. Basically, it was, of course, naive, and the sequencing of genome, been done, and so is other species, and we didn't find this master gene. Still, no one knows it. But what's interesting is the same species, right? Some of them live just a couple of weeks, others maybe 400 years, and mice live, as I said, just 2 years. And here we are, and you would think that we should have a different genome, but actually genome, as you could see, it's very similar. Like we have 85% similarity to homology to the mouse genome. But then why mice just live 2 years and we live 70, 80? So that kind of seems like, for me, didn't make a sense. And as I said, we still don't know what is it, the genetic factor or gene that tells you how long we live. Why not 200 years? But anyway, for us. So as you know, there is we live certain years because there is this aging. So for humans, I guess somewhere after 50, 60, everything goes down, and there are actually you could look it up. Of course, there is an aging phenotype. I mean, you don't have to be scientist to look at the person, say this and then this, and tell how old they are just by looking at them. And you don't have to even have a degree in biology. So the outside body can tell you your age. But nowadays we can actually look at the inside of a body in each cell, and then figure out that there's there is a damage in cells, and so it's called actually hallmarks, which is now popular, and there are so many different things can go wrong with the cell aging, and of course there are interventions being developed that supposed to extend our lifespan. But mostly I think it's not as much as just expanding lifespan, but making our, say, last 5, 10 years of our life when we aging and becoming old, be more, I guess, uh, uh, you know, enjoyable, as you know, because we all die of diseases. Um, and so I'm, as I said, I was always interested, these genetic factors, and that's why I was more interested saying what is it in the inside of aging cell is just pure consequence of aging, and I believe many of these hallmarks, it's just consequences, but there is I guess a root that actually causing it, and that's why I'm trying to study things that can actually prevent manifestation of many of this aging. So one thing that I have to say, so nowadays we can actually, if you can go back to cells, we can identify, you know, today's is by just looking at the cell or analyzing the age. Uh, Basically, you could send your DNA to, there are some commercial companies, and they can tell you how old are you, and which is no big deal. We actually don't need to know it, but anyway, there is the aging clock or sign in every cell. Um, but I was kind of very, surprised that, for example, not every cell in our body ages. As you know, we have eggs and sperm, and particularly say in adults, like 30 to 40 years old, every cell in their body have an aging clock, which tells you that this cell is say 30 to 40 years old. And of course, you could just look at the person, and you will know that this is not 20 years, it's not 60 years, but it's about 30 or 40. But you would think that the eggs and sperm will have the same age, and that means if you have a child, that child should live, shorter life, because the clock was already kind of 40 years when it came from a parents. But that's not the case, as you know. Children live normal lifespan; they don't have a shorter, right? So doesn't matter, from what age egg and sperm came from, particularly sperm. It could be from 80, 90 years old men, but the children have a normal lifespan. So that was one of the things that I was very much drawn to, and that's why my interest was in eggs and sperm, early embryos, to see what makes them rejuvenate or why they don't age. And that's the reason I started studying, the eggs and sperm and pre-implantation embryo, because I thought that's what the answer is, why we age and why some cells do not age. It's actually a pretty old theory, and one of the first scientists who noticed it was August Weismann. It was in 1892. He actually came up with this theory of germplasm and soma. So he actually noticed that the way that he says, he thought that there are, germplasm. Well, now we call it germ cells, and so he thought, and he was right, that they're immortal. So as you know, the gamete, genome of the gamete, and the egg and sperm, and they make an embryo. So this is kind of day 5 embryo. And what embryo does is actually contributes some cells into the soma. So it's a body cells, which are, all the organs, and basically it's our body. But a few cells are set aside during development, and they become precursors of the egg and sperm. And of course, Once the body become adult, these eggs and sperm develop into mature, and then they will be fertilized, and they keep going and going and going this cycle. So the body cells are mortal, and so they basically, that's what lifespan, what we know today is, let's say, somatic cells. It's 73.5. But germ cells are immortal, which is true. Like if you think that the genes that we carry, right? Of course, we got them from our parents. The parents got from our, grandparents, and so on. If you keep going backwards, you probably, if you trust, of course, theory of evolution, the genes that we have today, they probably came from our first living cells, some amoeba and so on. So that's where it is. It came without disruption, and we've got a lucky few that survived that long. But the body is of course, our somatic cells is disposable, and Weissman thought that the body is actually it's a house. So the germplasm builds body to protect itself, to live for one generation until it jumps to next and next and so on. So that's kind of one of his theory, that it's somatic cells have a limited lifespan, but germ cells are immortal. They never die, even though it's not completely true. The gametes actually terminally differentiated and they wouldn't leave. But after fertilization, something happens that they actually become rejuvenated. And that's why I started studying what is it that after fertilization makes them to live or add another lifespan. So one of the things Weissman noticed, it's called actually Weissman barrier, that once the embryo commits and puts cells during differentiation into somatic cells, they actually never will contribute to germ cells. The germ cells are kind of kept in this line, where somatic cells do not contribute to germ cells ever once they become separated, which is true. So it's called the Weissman barrier, which is, in nature, it doesn't, the somatic cells do not cross. But I just mentioned to you, right, we're trying to make sperm and egg from skin cells. So we're trying to break this natural barrier, and I'll tell you how we do that. So one of the way to do that is through cloning. So the cloning is another area besides just reproduction, where we see that how cloning is done. You take in this case skin cell or any other somatic cell, and it could come from any aged animal, say, or humans. It could be 80, 90 years humans. But we introduce it into cytoplasm of the donor egg. So we take donor egg from a young, say, woman or young animal, and that's how cloning works. So the first thing, the cloning actually showed that no matter how old your skin cell is or whatever we're using, all offspring produced, they live a lifespan normal, not at the age of somatic cells. And that's why, I become interested in this CNT. As you know, this is the dolly. It was in 98 that was first produced by Keith Campbell and Ian Wilmot. And since then, we cloned probably, more than 2,000 different mammals. And this is where frogs were first actually cloned in 1960s by John Gurdon. And actually, he was awarded a Nobel Prize for that. But more recently, I think about, I would say, 50 years after he made this discovery, because these frogs were produced in early 60s. But I think he got his Nobel Prize in 2012 or something like that. So, sometimes discoveries have to wait that long. But why he got it? Because of this, as I mentioned about this barrier, because somatic cells naturally in nature never cross this barrier. So they never become eggs and sperm and produce offspring. But the cloning made it possible. So anyway, as you know, I was interested in more doing the same procedure for primates, including humans. And so this is how cloning works. As I mentioned, we take any somatic cell nucleus. In this case, could be skin cells. That's our favorite. We call it fibroblasts, but it could be any other cell, as long as it's diploid. And and then we need an egg. Then, of course, egg comes from a donor, young, say, woman, 30 years old or so. So what we do is we remove from egg its own nucleus. It's actually spindle, we call it actually during meiotic cell division. But anyway, we remove it like it's shown in this pipette and discard it. So all we have, what's left is the just empty cytoplasm. I mean, it will look like a normal egg because it's a, nuclear DNA is tiny. So what we do is we take now this nucleus of the skin cell and we put it inside. So we replace the egg's nucleus with somatic cell. So that's what the cloning is, or CNT, somatic cell nuclear transfer, right? Because normally egg's nucleus is a haploid, as you know, it has a half set of chromosome, and that's why it needs sperm. But the skin cell, any other somatic cell, they have a diploid set. So we don't need sperm, and we cannot actually use sperm. If we bring sperm, we'll make a triploid embryo, which is not viable. But we learned actually to induce kind of activation. Usually sperm brings not only DNA, but actually triggers the egg to become embryo. And that trigger, it's called the kind of activation or egg activation. And that's actually one of the major problem in cloning, how to induce this activation without using sperm. But anyway, so if you succeed that, and that was one of the area of my expertise, now this egg with diploid nucleus becomes embryo. You remember that zygote, and of course it becomes now day 5 embryo. Of course, in humans we don't implant this, but in monkeys we can implant it, right, and make baby and cloned monkey, or sheep and so on. But in humans, what we do is with this embryo, you remember I told you we can actually establish embryonic stem cells, which is a kind of embryo cells, but now grow in a petri dish without differentiating, and you could actually expand them, millions of times, freeze thaw. But then you could actually try to differentiate them in vitro, and we have lots of techniques differentiating now this kind of early embryo. At this stage, there is no kind of specialized cells, but we can grow and differentiate, and then you could actually make in petri dish lots of different cell types, which is, well, first of all, tells you this is normal embryo. That means it's not just skin cell, because it becomes like normal embryo, because this is how embryonic stem cells would act. But most importantly, if you look at the now age and these cells, right, all of this cell doesn't matter, and you compare it to age of the original nucleus that we used, as I told you, right, this is actually can be 90 years old man or woman, doesn't matter. And now you look at these ones, it's just the same cell, but we kind of cycled through this nuclear transfer. Now if you look at them, they don't have age, they become like zero, right, just starting their life, not 90 years old. So that was one of the important things, that's why I'm interested in CNT. But also that's actually why, I'm using a CNT, not because I want to clone humans or animals, which is particularly in animal area, I guess it's a useful technology. But I was interested, so what makes reset of this aging? It was 90 years old, and now you have completely unborn cells, and that will have now another 90 years to live. Basically, we reset the lifespan. So that's the important question. Unfortunately, that's still there is no still answer, but I think I kind of believe that I found it. And one of the things that I study is the rejuvenating technology or mechanism is in the egg cytoplasm. This egg is its own DNA is removed. What's left is egg cytoplasm. So that's why I was drawn to the egg and more specifically egg cytoplasm, and what is there that makes everything in old cells go away and everything is reborn again. Naturally, we all, of course, we come from egg, right? And that's why we knew that there is something in egg, and it's not in sperm, and it's not in its DNA, right? It's in the cytoplasm. So anyway, that's how I came to become more focused on egg cytoplasm and to study what is it there. And one of the important things we call the maternal factors. That's been always there. That's what makes embryo to be reborn again, and that's why I'd say 40 years old parents will have a child which will live normal lifespan. Why? Well, because after fertilization, these egg factors, maternal factors, always induce rejuvenation. And the same happened if you put the somatic cells. So that's the whole idea. So the even more interesting is that if you do cloning in the mice, right? In this case, say this is adult mouse. As I mentioned, maximum lifespan in mice is 2 years. So say you have a 2 years old mice, very, very old. You take one skin cell, and of course, if you look at the skin cell, that doesn't matter what cells, they have a aging clock on them. It tells you there is so many things wrong with it. But if you do this nuclear transfer, you put nucleus inside of the cytoplasm of donor egg. Of course, egg has to come from young female. And bingo, you have rejuvenation. And this new embryo, we call it clone, right? Clone. It will live another lifespan. So these mice lived already 2 years, and we just took its old cell and we recycled it, and now the clone number 1 lives another 2 years. And that's actually not the only thing. Now you can take these 2 years, clone number 1, and make clone number 2, and so on and so on and so on. And one of my colleagues, he's Teryuka Wakayama in Japan, he's been working on this project since 98, I believe, when he first cloned mouse. He was the first to clone mice. And he is going with this, and I believe he has now, I think, so since 98, as you know, it's been 27 years. And he has that one mouse that was 2 years and 98. And now he keeps it going and going. And he says that somatic cell lifespan can be extended indefinitely because now he has probably more than 15 generations of these mice keep growing and growing. So again, that tells you there is something in egg that rejuvenates. But of course, each cycle of the cloning is a new egg. So each time eggs add only one lifespan, right? And then if you want to expand it again, you have to have another new egg. So that's the thing. One of the, as I said, the other things that the egg cytoplasm has, besides those maternal factors, is actually there is a genetic factor, and that's my interest in mitochondria and mtDNA. So that when we remove egg nucleus, right, it's all nucleus. Well, I draw it here very big. It's actually very tiny. So what's left in cytoplasm? As you know, there is, maternal factors. Those are kind of proteins, mRNA. But there is a genetic, the mitochondria that carry their own DNA. As you know, mitochondria, it's a cells inside of cells. Even though we think that the basic unit in our body is a cell, right? Maybe cardiomyocyte or neuron, but that's actually not true. The basic unit is a mitochondria, because mitochondria is not just the organelle inside of cell. It's cells because they have their own genome, kind of nucleus. And they, as you know, is very, similar to, one of the simple type of cells. And that's why I thought, there must be some genetic factor that contributes to the aging and lifespan. And it's been always known that it's a mitochondrial aging is one of the cornerstone of the aging. And here in the egg provides new mitochondria, and the skin cell provides just the DNA of the nucleus. Here you have a clone. So in clone, somatic cells contribute nuclear DNA, but we don't take the mitochondria. Somatic cells have the mitochondria, which we usually throw away, but mitochondria comes from egg, and that's, in my view, is a key why now we can rejuvenate this nucleus, because mitochondria comes new, fresh from the egg. And my theory is that it's a mitochondrial genome that actually defines our lifespan. So coming back to what's a mitochondria, it's actually very simple, organelle. I mean, the cell, I would say. The organelle is complicated, but it's a genome is pretty simple. And only 37 genes out of those 13 proteins, and majority of the genes that would require for, say, for replication, transcription, translation, they actually been transplanted to the nuclear genome, and it's considered to many years ago, actually more than 2 billion years ago, the mitochondria were free living, single cell organism. But then, our, I guess, precursor cell is also single cell. They join together and symbiosis, and that's how during evolution, I guess, majority of the mitochondrial genes, and used to be probably more than 1,000 genes there, they've been transported to the nucleus, but the 37 left there for whatever reason, and in all species. And still not clear why every gene hasn't been, transported to the nucleus. But anyway, so this is the mitochondrial genome, and even though majority of these genes actually responsible for producing of the energy, but one of the thing is that the mitochondrial genome is very prone to mutations. So this is actually one of the major pathways of how we produce energy. It's called oxidative phosphorylation. So we take basically, say, glucose molecule, and so before it goes to mitochondria, our cells can break it into by glycolysis, and only produces like a 2 ATP molecules, which is that's how our cells were producing energy before we got mitochondria inside of our cell. But this is a very inefficient way of producing energy. But mitochondria can take now these remaining, lactate and then pyruvates, and then make additional 28 molecules ATP. And you could see how efficient this energy is squeezing out of this glucose. And this is actually what believed to be contributed to major, evolutionary breakthrough. Up until then, we were single cell organisms, barely surviving because energy was kind of very scarce. Two ATP molecules from each glucose molecule. But once the mitochondria joined us, we were very rich. We could make now 2 plus 28, 30 ATP molecules. And of course, that was plenty of energy for cell to, basically to adapt and become multicellular organisms and so on. And that's how evolution just took off because of the energy. And the mitochondria, when they joined, as I said, they had not only the regulatory proteins encoded in them, but this oxidative phosphorylation or electron transport chain actually encoded by 5 complexes, and they are very, very complex proteins. And as you could see, there's say complex 1 encoded by 45 genes, which is a nucleus, but the 7 from mtDNA, and so it's complex 3, only 1. So there is these 13 proteins somehow left in mitochondria, where all these genes a long time ago, that means 2 billion years ago, they were actually in the mitochondria. But for whatever reason, they thought it can be go to nucleus, which as we know has a lifespan of limited. But these guys left there, and for whatever reason, these are immortal. So anyway, the other things to consider about the mitochondrial genome is its maternal inheritance. Only from egg. And they don't go through kind of recombination, and fertilization type, because it's called clonally propagated. So we clones of our moms in terms of mitochondria, and so on and so on. The other thing is we actually have 2 copies of each gene in the nucleus, maternal or paternal, but we get from mom thousand mtDNA copies. Well, egg, especially because it's large, has between 500,000 to 1 million mtDNA molecules. So that many genes. But why we need that many? But it's probably, because many of them we need to produce energy. But another thing is the mtDNA is very prone to mutations. It's considered about 100-fold more than nuclear genome. So during lifespan, we generate so many mutations, and that's one of the cornerstones why mitochondria age. But eggs provide us with no mutant mtDNA, and there is a theory why. Well, first of all, the mtDNA is, DNA in the mitochondria is not protected by histones, and that's why. And then another thing is mtDNA doesn't have a repair system like a nucleus, and so many other things makes it prone to mutations. But the interesting thing is that the eggs have a very protective system. So in germline, as I mentioned, it's also not clear why, but particularly probably because eggs do not use oxidative phosphorylation as source of energy, because you have to turn on the mitochondria, and the mitochondria will produce reactive oxygen species. That's actually major damaging thing, for mtDNA. So basically, DNA damage is the main thing that basically damages mtDNA, and that's why somatic cells gradually through age generate the mitochondrial DNA mutations. And of course, even though we have 1,000 copies, but mutation kind of accumulates and it catches up, and when say 60, 70, 80% of mtDNA in our cell mutant, so that the ability of cell produce energy declines, and that's what that aging phenotype will manifest. So we actually been studying somatic mitochondria, and we showed that there is a gradual accumulation of the somatic mtDNA mutations. And particularly, we looked at actually induced pluripotent cells. As you know, there is other way of making stem cells by directly reprogramming skin cells. But they will look like pluripotent cells. But the problem is that the older the skin cells are, basically the subjects that we make stem cells from, the more mutations. So anyway, this mitochondria, what makes all this lifespan to go. It's the first time, and from all mice, because this young mitochondria gives this old cell one lifespan, and then once it's expanded, now you take another, and then another, and so on, and that's why somatic cells could live forever. So I actually built this kind of concept of mitochondrial aging clock. As I mentioned, that we all come from egg, and it produces us with full range of mitochondria, and that means lifespan. But as we live, due to mutations, the energy production declines, and that's what contributes to aging. But as I mentioned, the egg cells will maintain normal mitochondria. Well, not forever, but up to like 35 years, 40. And then that's why children born will have normal lifespan. But then, mitochondria in eggs also start aging, but very drastically actually after 35. And that thing usually I study. But anyway, so if you make cloned stem cells, and in this case, you use mitochondria from young woman, then doesn't matter where the donor cells come. It could be from here, and we make normal embryonic stem cells, and their lifespan is normal. So they will have another full lifespan. And I mentioned to you about these iPS cells. I know there has been so much hype and still is. Oh, you can make now these iPS cells from old women and older patients and make them. But the problem is that yes, if it's from young or fetal or even adults, yeah, they may be. You could use it for transplantation and have young cells to replace your aged. But the problem is that majority of the diseases start at this age, and these mitochondria, because they use their own mitochondria, aged mitochondria, so they actually have very limited ability to expand our life. So that's my concept, and I mentioned to you that just to show that this is a real thing, we can replace mitochondria in older women. As I mentioned, IVF, if it use their own eggs, only works like up until 35. After that, there is a drastic decline in our ability to produce babies through IVF. But we know that if these women take donor egg completely, not integrated, they can produce babies in up to their 50s. So that's why we think that it's due to mitochondrial aging, and we developed a procedure. It's called mitochondrial replacement therapy, as I remember. So we take patient, say in this case, 45, 50 years old, and we remove its spindle nuclear material, but we throw away mitochondria in cytoplasm. But we take a donor egg from young donor, they 25, 30 years old, and then we remove the nucleus and we basically replace it. So it's a kind of nuclear transfer procedure. We call it spindle transfer. And all suddenly now you can rejuvenate this egg from the patients, because we retain the nuclear genome. But mitochondria, of course, comes from a donor egg, and we produced many papers and macaques, but most importantly, this actually, it's in the clinics now. So this is our, a few years ago, we finished this clinical trial when we did for women. Well, this case, they were not that old, they under 40, but they were infertile because they went through many regular IVF cycles with their own eggs, and actually 6 on average. And they couldn't become pregnant, but we used this MRT, only 1 cycle, and we had 6 babies born, which is 24% of live birth rate, which is pretty high considering that they had a 0 before. So that's one of the proof of concept when transferring young mitochondria from unaffected egg can, aging of the cell go away. So the idea is that the egg is only one of the 200 or so cells in our body. Of course, it ages very early. By 35, it starts aging. But our other cell types, as you know, they catch up a little bit later, and the aging phenotype, and particularly the aging mechanism is the same. It's mitochondrial aging. And the idea is, can we use now young mitochondrial transfer to eliminate age-related diseases? Of course, not to make them just young, but at least not to suffer from many of these diseases. And that's what my last, I guess, 5-6 years of the research was towards of expanding this mitochondrial transfer rejuvenation from just one cell, which is egg, to other 230 or so cell types. So it's more difficult. It's the way we do it is difficult. And then also technology is pretty going to be difficult, but that seems like doable. And so we just think we'll keep working. And unfortunately, it may not be that quick process. Who knows? It will take maybe 10-20 years. But I have a really big lab, but many of them actually graduated from my lab as a postdocs, and they continue with their own labs, and most of them hopefully will stay in this area of mitochondrial rejuvenation and carry these projects towards. So hopefully we'll develop these treatments someday. But these are just my today's lab members. You could see not a lot. But I think that's all. I'm sorry, I think I took a little bit longer probably than I should.
Thank you so much, Professor Mitalipov, for the fascinating and insightful talk, sharing about your background and some details on and the concepts of your idea. It was really, really interesting, and we received many questions, both regarding the science itself and also about the career advice. And let's address them in the Q&A session. I would like to start with biological questions. Taisia asked, she's curious about the techniques. How do you deliver gene therapy systems? And do you use methods involving nanocarriers or work with liposomes, inorganic nanoparticles, especially nanosilver?
Yes, so to do the gene corrections, I guess, or CRISPR, for example, system in early eggs or embryos, luckily we don't need any additional, like a system to deliver. We basically just inject. And we have a tiny pipettes, and we can inject them directly into cytoplasm or even in pronucleus. But more simple, as you know, nowadays we do fertilization by injecting sperm, right, directly into the egg. It's called intracytoplasmic sperm injection, and there is a kind of short abbreviation. It's called ICSI. I don't know why ICSI, but so during ICSI we inject sperm, and what we do is we kind of pick up the guide RNA and the CRISPR system with tiny amount of fluid and sperm, and we just co-inject. So it's pretty simple. So delivery, luckily for embryos, has never been a problem. We don't need any viruses or other system or electroporation.
Can I ask a following question regards from this part? Like, and how you screen, for example, for an off-target effects in this case, if it's a direct incorporation, right, basically, so, and it's not an in vitro system?
Say it again?
How do you screen for the off-target effects? So when you use the CRISPR and the guide, right, basically, so we have a lot of off-target effects, and this is like so it's not like an in vitro system if you're gonna like implant this directly.
Yeah, so yeah, of course, the first thing is, for example, before we apply on directly on embryos, we of course we test every guide RNA and then system with the cultured cells. For example, we take skin cell and we get the fibroblast culture from a patient. And then we target them, with biopreparation, and that's where we look at the off target. So we test on and off target in cultured cells, and only after that we inject them into embryo during fertilization. But then, we have a window of 5 days culture when this embryo becomes blast cyst. And as you know, we can take a biopsy of the trifectoderm, which is pretty standard nowadays for pre-implantation genetic testing because it's done actually with every IVF embryo nowadays. And that biopsy can be now amplified, and we can look at the on and off target issues with that biopsy. And then if that particular embryo looks good, that means we did it really correction as we wanted, and there is no off target effects, then I guess we can transfer that embryo.
Okay, thank you so much.
Thank you, Diana,
Even
for
though,
your question.
it's not really allowed yet. So we can do in vitro, particularly with humans, but we don't implant to transplant yet because the system is still, as you mentioned, there is still issues with safety. But unfortunately, there are some IVF clinics where they think it's okay. They just inject and implant. And as you know, there's been a case in China. But that guy who did it, he went to jail. Yes. So that's a warning. Do not do that. Particularly, I tell them to all IVF clinics, you guys gonna go to jail if you. It's not like they know what they're doing, but even if they can copy paste, guide RNA, that's not allowable yet.
Just like so when you're doing the mitochondrial gene transfer, right? So I've heard that UK and Australia, like they're allowed to do the clinical translation, right? I'm not fully into the legal part, but what is the benefit that allowed those countries basically to go and proceed rather than not Germany nor US as well cannot move forward with that?
Yeah, I think it's mostly politics. Yeah, with mitochondrial gene therapy, as I mentioned, we don't use the gene correction, neither CRISPR system, because there is too many of them, 500,000 copies. And that's, all gene correction techniques are based on DNA self-repair, but mtDNA doesn't go through the repair. And that's why the best way there is remove mutant mtDNA in egg and replace it with a donor. So that's called mitochondrial DNA replacement, which is actually safe because we tested it in, more than 15 years ago in macaques, and then we produced so many mice, and then we tested with human embryos without transplanting. And that's why I think we wanted to do clinical trials. But unfortunately, in the US, it hasn't been allowed. Mostly it's because of the political system, political issues that they think that, this is, gene transfer and then gene therapy that unproven. But UK and Australia, they were first to approve. Actually approached many regulatory agencies in different countries. So, Keith Campbell, actually, he's my friend. He was actually a member of the HFEA in UK. So that's how I know he helped us to go through review, and those countries allowed it. Yeah.
And just sorry for everyone, and just as a last question, because I'm myself a geneticist, why are you skeptical about the human IPCs?
Because of the aging phenotype in them. You can rejuvenate, I guess, it's epigenetic rejuvenation of the nucleus, but you cannot rejuvenate mitochondria because, iPS cells have the same mitochondria from, say, 80, 90 years old patients. And that part has been shown, and we showed that they have lots of mutations, and the somatic mutations, they not inherited, acquired. And we say, unless you can correct them, which is impossible at this point, or you have to replace it. And that, again, that becomes, where are you going to take donor mitochondria? And they have to come from egg. That's only unaffected source of mtDNA I found. It cannot be any somatic, mtDNA. It has to be germline if you want to produce normal with full lifespan cell type.
Thank you, Diana, for your questions. And I would like to move on to a different kind of topic of questions. We have some questions about career advice. Did you do anything particularly different during your last year of PhD or during your postdoc to kind of set the start of your kind of success or the progress of your research? Would you give any advice for people in that stage of their career?
Well, yeah, I mean, particularly when I was finishing my PhD, it was mid 90s. It was actually worst time probably to go to science because particularly in Soviet Union, everything was going south and there was no funding and so on. So that's actually prompted me. You either have to go and I don't know, start selling in Baracholka like all my friends did, or try to immigrate and get the going on with the science. So I guess one of the issues after you finish your PhD is you have to go through, I guess, postdoctoral training. So there is a certain stages we have to go through. Unfortunately, even in US and most countries, the our salaries or stipends are very low, and that's why no Americans would go to go postdoc because it's just. But for us, I guess it's still you have to be I guess really trusting in science. So you give up I guess idea of being you can earn something. So you will be always poor, I guess, at least in the beginning of your career. The other thing is, one of the key in at least in United States, key in becoming independent scientist, right? Not like working for somebody, is to be able to get your own funding. As you know, there is things in here, I guess, in capitalism. It's not like somebody gives me money or university or something. No, money you have to earn, bring it. You have to win it through the grants and so on. So that's a very hard time. The moment you can have your own grant, that means pay your salary, but that's small part. You have to pay for salary of every member of your lab, which is probably half of your, and then the other half have to, for eggs, for all the reagents, and you have to pay for, lab space, and then you have to pay for equipment. So that's what makes PI, so it means principal investigator. And that part is actually very, very difficult. And especially, coming from Russia or somewhere, we never thought of. You thought, oh, there's going to be a clot, that you're going to be able. No, you have to be able to live on your own. So that's actually very, I think, the key. If you're successful in that, you become independent. And this, at least here in the US, we freelancers, right? I work in university, yeah, but it's not like somebody, my boss tells me what to do, what to study, or how to do my science. No, it's absolutely my idea. If I decide tomorrow I don't want to study mitochondria, maybe I want to do something else, it's up to me. I could just do whatever I want to do, as long as I have money, my own money to do it. And that's the difficult part. And how you get this money to run everything. In science, we don't produce something you could sell, right? All we produce is, discoveries, maybe papers. So that means you have to know that the product is scientific discovery. It's a papers, it's impact factors. And that's what you can use to sell to agencies that give grants. And of course, the science you produce is already in the past, but when you're asking money for something that hasn't been discovered, so it's a proposal. You're saying, if I do this and this, I'm going to have greater discovery. This is going to implicate so much in this area or this area, and I will produce additional nature science papers. And that's what you have to convince, say, NIH here in US to get money. So it's a kind of, circle. To have to be able to produce papers, you have to have funding, but you have to have, past, I guess, publication record to be able to apply for grant and win. Because, many agencies give my money like in National Institute of Health here. But I think at least now, only maybe 5 to 7% top grants win. So that's a pay line. That means more than 90% grants are not going to be funded. So you don't want to be on this 90%. You want to be on this top 5% to be able to compete for grants. And most of the time, you cannot have one grant because grants are small. You have to have many of them. And you have to have millions and millions of dollars. You have to be a millionaire to be able to fund your lab, especially say, how many people in your lab? Well, you could have 100 or 1,000, or you could have just one, or just yourself. It depends on how successful you are with getting money. And that part, unfortunately, is difficult. I don't think it's they teach you in PhD schools. Yeah, maybe they teach you, oh yeah, this is how you apply for grants. There is the kind of structure. But this is a system how it works, I guess. I had to learn it because it took me a while before I got my first grant. And then it was a while before I had my second grant. So that's I guess, tough part of the being scientist.
The trial and error, just trying to improve your research and then your application documents and grant application documents, right?
And meanwhile, you have to keep producing, you have to have a good papers. Otherwise, without good publication prior record, you're not going to be in that top 5 percentile, I guess.
Thank you so much, Professor. We would like to wrap up this Q&A session. Your insightful talk sparked curiosity among people who are bio-major and then people who are not bio-major, and it's very inspiring to see. Would it be okay if we send you some questions, remaining questions, to get answers so that our audience can get the responses? To finish this event, we would like to know a little bit about your startup company, the Mitogenome Therapeutics. Maybe if you can share your current status and future directions, that would be a nice kind of wrap up of this event.
Yeah, overall, this West Coast like culture of especially Silicon Valley, I think, is all areas where you live, how it's shaped you as a scientist, and you built your startup company. It's very interesting for us. Thank you.
Yeah, I think I primarily was, as I said, the academic professor, and all the money I was earning, it was grants, basically from government, or usually there is non-profit foundations, they fund, gives grant. But with this aging, I guess, concept, as you know, there is a renowned interest in rejuvenation and life extension, and that's how, because I was interested in, as I said, in aging, particularly with this at the last I guess my slide was on using this mitochondrial rejuvenation to treat many age-related diseases, not just in eggs, but say maybe in the brain and heart. So that's how this interest came of. There is a interest in venture capital to invest in scientific research, and that's what the mitogenome is for. So primarily the money we generate through venture capitalism. So basically, they will have a share of the company, but the money I will invest primarily into my center, my lab, maybe some other labs of my graduates. But the idea is that the discoveries that we make, intellectual property, which we usually make, that's actually a side part of the, we make lots of patents, but normally they belong to university because that's the, system that works. But in this case, because it's a company and it's venture capital, so we invest into the science, but through sponsored research agreements. and we will have agreement with the university saying, okay, I'll invest, say, this $10 million or $20 million into this project, but in return, we want to own intellectual property. We will own patents. So the company, Mighty Genome, will own. And of course, whoever has shares will co-own this technology. And the idea is that they will keep going, and then in the future, I guess that's how we're gonna make money by either selling the whole package of intellectual property or continuing with these treatments and maybe building I guess therapies and then making money by selling these therapies. So that's a whole idea behind these startups, and in this case the mitogenome therapeutics is towards of delivering young donor, it's actually oocyte mitochondria, into aged neurons, heart cells, and any other cell type, with the idea of treating age onset conditions, but also extending healthy human lifespan.
Thank you, Professor Mitalipov, for your fascinating talk. We wish you success in your future research, and we will follow closely the progress. And I would like to thank everyone who joined this session and for your questions, for your interest, curiosity. On behalf of the Kazakh International Science and Technology Association, I would like to express our gratitude, and I hope to see you again in our future events. Thank you so much.
Thank you too, and thank you again for invitation. And if there is more questions from audience, I guess you could direct to my either email or I'll try to respond either personally or maybe through your forum.
Thank you so
Thank you.
much.
Bye.
Bye bye.